Ever wondered how the blueprint of life translates into the myriad forms of life we see around us? This transformation is orchestrated by a fundamental framework called the Central Dogma of molecular biology. This concept, proposed by the brilliant mind of Francis Crick in 1958, governs the flow of genetic information within every living organism. It's like the conductor of an orchestra guiding the symphony of life. At the heart of this grand concert are three main acts: replication, transcription, and translation. They're like the musicians playing their parts, each contributing to the overall performance. They ensure that the music, or in this case the genetic information, flows seamlessly from the DNA, which is the score, to the RNA, the conductor's interpretation, and finally, to the protein, the performance itself. So buckle up as we embark on this fascinating journey of genetic information, from DNA to RNA to protein. Our journey begins with replication, the process which ensures life's continuity. Think about it as the grand photocopy machine of life, duplicating the intricate genetic script embedded in our DNA. This duplication is the starting point in the central dogma of molecular biology, a process that guarantees that each new cell receives a complete set of genetic instructions. Imagine a twisted ladder, that's our DNA. The first step in DNA replication involves the careful unwinding of this double helix structure. This is akin to unzipping a zipper, where the two strands of the DNA ladder are separated to expose the rungs made of pairs of nucleotide bases. These bases, adenine, guanine, cytosine, and thymine, are the fundamental letters of the genetic alphabet. Once the DNA is unzipped, the real magic begins. New complementary strands are synthesized or built, one nucleotide at a time. This is done by matching the exposed bases on the original strand with their corresponding partners, adenine with thymine and cytosine with guanine. This dance of base pairing forms a new DNA molecule that is a mirror image of the original. But wait, our cells are not just good at copying, they are also meticulous proofreaders. As the new DNA strands are synthesized, the cells constantly check for errors or mispairing of bases. If detected, these errors are promptly corrected. This proofreading and error correction mechanism ensures genomic integrity, maintaining the accuracy of the genetic information that is passed on to the new cells. In essence, replication is a beautifully choreographed biological process that safeguards the continuity of life. It ensures that the genetic information, the life's blueprint, is faithfully duplicated. This process is not just a simple copy-paste job, but rather a complex symphony of unwinding, synthesizing, and proofreading, all working seamlessly to produce an identical copy of DNA. So that's how every cell gets an identical copy of the DNA. Next, let's see how this DNA blueprint is put to work. Now that we have an identical copy of DNA, it's time to put this information into action through transcription. Imagine transcription as a diligent scribe meticulously copying a segment of DNA onto a complementary RNA molecule. This process is akin to a skilled artist rendering a detailed sketch from a complex photograph. The artisan in this case is the enzyme RNA polymerase. It's the driving force behind the transcription process, turning our DNA blueprint into a messenger RNA, or mRNA, which will carry our genetic message out of the nucleus and into the cytoplasm. Let's delve a bit deeper into this fascinating process. First, RNA polymerase has to recognize and bind to a specific DNA sequence. Think of this as the starting point of our sketch. This sequence is known as the promoter region, and it's the go signal for the enzyme to start transcribing. Once RNA polymerase binds to the promoter region, it kickstarts the process of RNA synthesis. The DNA double helix unwinds, and the RNA polymerase starts to assemble a chain of RNA. This chain is a mirror reflection of the DNA sequence, a complementary code. As RNA polymerase moves along the DNA strand, it continues to elongate the RNA transcript. This step is like the artist filling in the details of the sketch, ensuring every line and every shade is accurately represented. But every good artist knows when to stop. The same principle applies to RNA polymerase. When it encounters a specific sequence of DNA known as the terminator, it knows it's time to stop transcribing, to lift the pen off the paper. The RNA molecule is then released, and the DNA helix zips back up. And there we have it, a messenger ready to deliver the genetic information to the cytoplasm. But how does this message get deciphered? Well, that's a tale for the next part of our journey, where we explore the fascinating process of translation. Stay tuned as we continue to unravel the central dogma of molecular biology. With our messenger RNA in hand, it's time to decode its message through translation. But what does this mean exactly? Well, translation is the final and perhaps the most fascinating step of the central dogma. Here, the genetic information carried by the messenger RNA, or mRNA, is decoded into a language that cells can use to build proteins. This process takes place on tiny cellular factories known as ribosomes. Imagine ribosomes as master chefs who have been handed a recipe, the mRNA, and now must assemble the ingredients, the amino acids, to create a delicious dish, a protein. But these chefs need assistance, and that's where transfer RNA, or tRNA, comes into play. tRNA acts like a delivery truck, bringing the right amino acids to the ribosome according to the sequence of codons, or three-letter words, on the mRNA. Each tRNA molecule has a specific amino acid attached to it and an anticodon that matches a codon on the mRNA. It's a bit like a lock and key mechanism. The ribosome reads the mRNA sequence, codon by codon. And as each tRNA molecule comes along with the correct anticodon, it adds its amino acid to the growing chain. This sequential addition of amino acids forms a long chain known as a polypeptide. But we're not finished yet. This long, linear polypeptide chain needs to fold into a specific three-dimensional shape to become a functional protein. Think of it as origami at a molecular level. The shape a protein takes is crucial to its function. Whether it becomes an enzyme to speed up chemical reactions, a hormone to send messages, or an antibody to fight off invaders, it's all determined by how it folds. So, from a simple DNA sequence through transcription and translation, we finally have a complex, functional protein. A remarkable journey, isn't it? And voila. From a simple DNA sequence, we have a complex, functional protein. But is this the whole story? Stay tuned, because there's more to the central dogma than meets the eye. The central dogma provides the basic blueprint, but life, as we know, is full of exceptions. In our journey through the central dogma, we've discovered how life's intricate machinery runs on the genetic information encoded in DNA, transcribed into RNA, and finally translated into proteins. Yet, the complexity of life extends beyond this fundamental framework, presenting intriguing exceptions and nuances. Take, for instance, the case of certain viruses. These crafty invaders employ a process called reverse transcription to literally flip the script on the central dogma. Instead of the usual DNA to RNA flow, reverse transcription involves the synthesis of DNA from an RNA template. This crafty maneuver allows these viruses, like the infamous HIV, to integrate their genetic material into the host's DNA, effectively hijacking the host's cellular machinery for their own replication. But the surprises don't stop there. The central dogma's linear model, from DNA to RNA to protein, has been expanded to accommodate the multifaceted roles of RNA. Enter the realm of non-coding RNAs. Contrary to the messenger RNAs we've discussed, these RNAs do not code for proteins. Instead, they play integral roles in regulating gene expression, controlling the when, where, and how much of protein production. This regulatory role adds another layer of complexity to the genetic landscape, underlining the intricacy of life's processes. Moreover, recent advancements in molecular biology have revealed that the central dogma is not as rigid as once thought. Epigenetic modifications, for example, can alter the way genes are read by the machinery of the cell, influencing the outcome without changing the DNA sequence itself. So, in essence, the central dogma provides us with a bedrock understanding of genetic processes, a fundamental guide to the flow of genetic information. But, like any good scientific theory, it's a launching pad for further exploration, a framework to be built upon, refined, and sometimes turned on its head. So, while we've unraveled the basic blueprint of life, there's still much to explore in the fascinating world of genetics. Let's delve deeper into the captivating interplay between genotype and phenotype. This is another dimension of the genetic landscape that the central dogma helps us understand. The genotype, or the genetic makeup of an organism, and the phenotype, the observable traits, are intrinsically linked. Variations in DNA sequences can give rise to diverse traits and characteristics observed in living organisms. Mutations, for instance, can introduce new traits, while genetic recombination can mix and match existing ones. These processes contribute to genetic diversity, the raw material for evolution. They form the basis for the breathtaking array of life forms we see around us. From the simplest bacteria to the most complex mammals, each unique and yet built on the same fundamental principles of genetics. Epigenetic modifications, as we mentioned earlier, add another layer of complexity. They can subtly tweak how genetic information is read and processed, influencing traits without changing the DNA sequence itself. This can result in identical twins having different susceptibilities to diseases, or even the vibrant colors of a peacock's feathers. In essence, the Central Dogma provides a roadmap to understand how the static, unchanging DNA can give rise to the dynamic, ever-changing world of traits and phenotypes. It underscores the incredible complexity and versatility of life, and how the same set of rules can lead to an astounding diversity. As we continue to explore the world of genetics, the Central Dogma remains our guiding light, illuminating the path to unraveling life's many mysteries.